promoter molecule that covers over the gene when it's plugged up. When you use up that molecule, the promoter opens back up again so the gene can be expressed. So the site of regulation of gene expression is actually the promoter. It happens at the promoter. And they'll call it a repressor protein in your book. Okay, what do we call it when a gene is copied into RNA? Transcription, right? Gene cannot be transcribed. Can't make mRNA, don't have mRNA, can't make the protein. That's a feedback inhibition. Actually happens at the promoter. The promoter is actually the site of regulation of gene expression. What's the terminator? Ends the gene. That's all it does. Remember that for your test. Terminator ends the gene. Why is that helpful? Tells you when your mRNA is done, right? Also stops frame shift mutations from running rampant throughout the whole genome. Let's go back to this. Okay, you inhibited your gene expression because the molecule of end product plugged up the promoter, causing the promoter to block the gene. Once you use up the end product, it unplugs the promoter because you're going to use up the very molecule of end product that's plugging up the promoter. The promoter is going to release the gene and let it go back to work to make lactase again. So the inhibition is removed once your end product is used up. Okay, now let's do induction. Which is the molecule they use for induction in your textbook. Okay, let's say I'm a little bacterial cell living on a dairy farm. Usually have lactose for supper. So I make lactase enzyme frequently. And all of a sudden, sloppy farmer slips in the cow patty and kicks me all the way onto a pile of hay that's going all the way to South America. And I get transported to this new environment. And in this new environment, guess what? It's not a dairy farm. It's a cattle farm. These are not cattle for milking. I don't get any milk. These are cattle for steaks. Now all of a sudden I'm going to have to eat protein. So instead of making lactase, it's not going to help me. I need to make a new enzyme, protease perhaps. So I better go back to my recipe book, my genome, and see if I got any genes for protease. Oh, look at that. One of my ancestors had a mutation. Sure enough, I have a protease recipe here. So the presence of a new nutrient may induce a gene to be expressed. So induction is turning on a gene that's not normally expressed. what would make you do that? The presence of a new nutrient, which we call an inducer. So it's a positive feedback. The positive feedback, the positive signal, is the presence of a new nutrient. The inducer will bind to a promoter on a different gene, the gene that codes for the new protein, and cause that promoter to open. Let the gene make the enzyme to digest the nutrient. Once the nutrient is digested and gone, the promoter will cover the gene back up so that it's not making the enzyme to digest the new nutrient anymore. So, don't think of a light switch. It's not the same room on and off. It's two different rooms. So let's say this room we use frequently. Every day I come in, I turn the lights on. At the end of the day, I turn the lights off. That room next door, you're not even aware that room is there. It's so small, we never use it. Now all of a sudden we're going to have a new class. It's a small class, 6 students in it. Where are we going to put them? Well, we're going to use that room. We never used that room before because we never offered this class before. It's a small 6-student writing class. Now all of a sudden, we're going to turn the light on. It's normally off, but we're going to turn it on. This room, it's normally on. At the end of the day, I turn it off. And then the next day, it's on again because I have more students coming. So it's 2 different genes. Some genes are normally open. In repression, you're going to close them. Other genes are normally off. You don't use them. It's a gene you almost never use. But for some reason, you might have to use it now. New environment, new situation, new nutrient. You're going to have to turn on the gene and start making enzymes to live in your new environment. That's all I can tell you. That's the way it is. Okay, how about the bacterial growth curve? Remember the lag phase? What are the cells doing? They're acclimating. So here's the bacteria living in poop. They turn on the enzymes for digesting poop. They always use those enzymes because they're always living in poop and they always have to digest poop. Until Dr. Pasteur goes out and takes them out of the poop and puts them in a test tube of nutrient broth. Now, they're not going to be using those enzymes that they were using before. Stop making those. I have no reason to. And now the presence of this new nutrient broth, I hope I have a gene to help me digest it. If I'm lucky, I do. Some might not have it. What's going to happen to them in the new environment? They're going to die of starvation, exactly. So that's where mutations might be a good thing. A mutation might result in a gene that maybe most of the population doesn't even use in the current environment, but as the environment changes, the ones that have that gene will survive. And the ones that don't have it won't survive. And this is how organisms adapt to their new environment and how populations change over time as the environment changes, and that's natural selection and evolution and all that stuff. Okay, for your tests, real easy. Repression, turn off a gene that's normally on. Induction, turn on a gene that's normally off. So promoters on different genes, some promoters keep the gene closed most of the time. Other promoters keep the gene open most of the time. But either way, the promoter is what's regulating the gene expression. The terminator tells the cell, if you're going to make an mRNA copy of this gene, start at the beginning where the promoter is and stop at the terminator. So the promoter is not only the site of regulation of gene expression, but it also signals the start of the gene. So the gene, the DNA, has a start and a stop, a promoter and a terminator. When you're making mRNA, that's where you have start codons and stop codons. So the mRNA knows where to start being translated and stop being translated, where it starts being transcribed and stops being transcribed. All right. Got a little bit of time left, so let me see what we got here. Okay, this is lecture material that is actually more covered in what we're doing in lab today. Is it opening? Okay, this is chapter 9. Also in your handouts package, you have some definitions of antiseptic, disinfection, sterilization, sanitization, pasteurization. You need to know the definition of those terms. Real easy. Sanitization, I wiped it off.